Okay, dear colleagues, when I started yesterday with my talk, I said that this conference is also a beat in the chain of the 10-year FAANG anniversary. And to show this and to show also how we clustered and all the interconnections we had with these Horizon 2020 projects, I invited the other Horizon 2020 project to give a brief overview of also their activities. And I was really lucky that they agreed to give a short presentation about what they had, and probably you will also see how often it's interconnected with BOVREC and with the other projects. So the start is about AQUA-FAANG. It's about the aquaculture. So they have very specific challenges with, for example, having more than one species, and also the starting point probably was much more challenging than we had with previous data on cattle already available.
Thanks a lot, Krista, for inviting me and to present the AQUA-FAANG project. So my name is Sigbjørn Lien. I'm from the Norwegian University of Life Sciences, south of Oslo in Norway. And you probably know that we are doing a lot of salmon production in Norway, so I think that was partly why I was very much involved in starting to doing functional genomics in salmon. I have a time some years ago working in cattle genomics, so I kind of did a shift at some point. Yeah. Yes, so AQUA-FAANG is, like Krista said, is kind of a sister project of BOVREC and also GeneSwitch. So these three projects were funded under the same call, and we started AQUA-FAANG in May 2019, and it ran for 4.5 years. And so we got a kind of a half-year COVID extension, and you have probably had that as well. So for us, it was needed because we had quite a distributed model for doing the analysis in many labs, and many labs were closed down, and that was quite a problem for us. I coordinated this project together with Dan McQueen at Russell Institute, and we have a lot of partners. I will come back to them later. Yes, so one of the challenges we have is not working, that we have been not working on one species, but we targeted 6 species that we consider to be the most important for European aquaculture. So Atlantic salmon, rainbow trout, sea bass, sea bream, turbot, and carp. So kind of developing the tools for 6 different species, and we wanted to do this in a standardized way so we could compare data across species, across tissues, and at different stages. Let's go to the next slide. Just have a background from aquaculture, and this is just to link in with what you are doing in cattle and other livestock species. So a lot of the tools, genomic selection, has been implemented in some of the aquaculture species, but not necessarily all of them. The most advanced industries, that's the salmon industry that we have a lot of in Norway and also in the UK. And what we have in those species, like in salmon, it's kind of unique. We have these industries were started in the late 60s for salmon, for example, and we have still quite a lot of major genes segregating in these populations. We haven't had the time to fixate some large effect loci. So still there are major QTLs segregating in the populations, especially for diseases. And this success story with the IPN locus, so a locus that is explaining 80% of the genetic variation for one disease, that has been really a success story, and a kind of a story driving the use of genomics and genomic selection in aquaculture. But now we have also problems facing the industry, and we see that genomic selection is working well with a set of SNPs. The industries are using normally 50 to 60K SNP arrays like you do probably also in cattle and pigs. And it works well. You don't even need 50K. You can do this with a LD set of SNP markers, let's say 3K or 5K. If you do it with close relatives, so you have one set of animals where you challenge with diseases, and then you do the correlation between markers and the disease, and you apply that in the broader population. So this works well, but the problem is that it doesn't work well if you want to do a cross population, so different year classes of salmon, and it doesn't work well if you want to do this across generations. So that's a problem in aquaculture, because doing a lot of disease challenges is costly, yes, but it's also very negative for animal welfare. And there is a threat that this will be forbidden to do in the future, because it's really not good practice. Okay, so this is challenging for the industry, because we need to find new solutions to this. And that's why maybe the functional annotation could support this by maybe we can find the underlying genetic variation making the difference in the different phenotypes, and especially focusing on diseases. So the project has been focusing on five main project objectives. So the first part is to develop assays. So starting with assays from humans, but adapt them to 6 different species, to different tissues, to developmental stages. So quite a lot of challenges related to that. And we also wanted to develop some tools to challenge fish, so make them kind of ways of challenge in vitro and in vivo. So something we could develop for further use. And use the data to predict possibly diseases in aquaculture. And also we have included in the study 6 different species. Maybe we can produce data that could be applied across species. We have 2 species with duplicated genomes, 3 species with duplicated genomes, making new challenges, but also very interesting. Okay, so these are the data we have produced, 6 different species. I have already mentioned them. We developed assays for attack and ChIP-seq for histone modifications and RNA-seq, which is quite trivial to do. We developed this for body maps, so 5 different tissues: liver, brain, gill, muscle, and ovary, testis, in immature and mature fish. And we developed something we call DEMMAP, which is embryonic stages from blastula to post-segmentation in all 6 species. And we developed some standardized stimulations or mimics of immune response using poly-IC and bacterial mimics, both in vivo and in vitro. And all this data is quite an amazing set of data. We have made all the data available through Ensemble, including the metadata, so documenting when samples were sampled and make everything public. And I think for the long-term use of this is very important. So this is a resource of data that can be used in studies after AQUA-FAANG. And having that kind of standardized in a proper way is very, very important for the future. And I also want to mention that the development of the assays is not done in one lab in isolation. So we have kind of trained multiple labs in Europe, from Norway to Italy, Spain, Greece, France, UK, Poland, to train them to use these technologies and do it in a standardized way so we can compare data across studies. And I think also in the future this will be very important. And we see already that people are using these assays in new studies. Okay, so this is for the kind of how we can use this data. It's not so easy to see, but this is what we do with the data is that we have the data from body map, the map, the disease challenge data, and then we overlay them with genetic variation, and that is normally information from the breeding companies or producers, so do genotyping in their populations. For example, assays from whole genome sequencing, or it could also be from SNP array data. And then kind of estimate what could be the effect of those regions that we identify as regulatory regions in the genome, identify the variation in those regions, and see if we can use that for developing assays for them. For some industries, it's quite interesting to use a subset of markers. For other industries, like they want to enrich the SNP arrays with functional data, and also maybe targets for genome editing in the future. So kind of already touched upon in the previous presentations in BOVREG. What we also have started to do, not directly in AQUA-FAANG, but in Norway and also other labs in the consortium, is to use deep learning to predict functionality of SNP variation within regulatory region. And we find that very interesting and quite successful when it comes to giving a prediction of what kind of variation within regulatory region are really making a difference. So that's potentially also something, maybe you have looked into that as well in the bovine community. So in the future, we see this as a further development. We are now seeing this being developed from SNPs and whole genome sequencing with short reads, which is detecting only a small fraction of the DNA variation, the SNP variation, and is being expanded to pangenomes, long read sequencing, and very much into structural variation. And in the future, I think we will see this expand with implementing, also getting that information into the equation and try to understand the functionality of structural variation. We also see quite a lot of modeling of identifying the transcription binding sites in the sequence data, and also conserved elements in the genome. All that, and also results from other studies, EQTL and GTX studies. But these are not really included in the AQUA-FAANG project, but we see this developing. So all these kind of new technologies and possibilities using functional data, we have developed this AQUA-FAANG white paper that came out in Aquaculture. So kind of try to make this attractive for the community, those people working in aquaculture. I think I don't have time to go through this, but this is just illustrate how we can use the functional variation. So prioritizing SNPs, often we find a lot of SNPs within a region. What SNPs within that region could be functional? You can overlay it with the functional data and then prioritize them, put them on a SNP array. So this is for fillet color, so where we have a number of QTLs segregating in Atlantic salmon, and illustrates how, okay, here is a QTL. What is the functional variation in this region? Is it within the BCO1 gene or the BCO1-like gene, or what kind of variation within the region? Is it regulatory? Is it coding? And so forth. So we strongly believe that AQUA-FAANG has created a large upstep in farm fish functional genomics, but we also see that the industries, so the users of this technology, or the whole industry is quite diverse when it comes to uptake of the technology. We have very advanced industries, big breeding companies in salmon, for example, that they are really at the level where they use this information. And they are also very interested in gene editing and using new technologies. But in other species, there are many species that are going into aquaculture. We don't have these really advanced industries, and it's more like using more traditional tools to improve the efficiency and the health of the animals. Okay, so this is just to summarize AQUA-FAANG objectives. I think we succeeded to make functional assays across species and tissues, and so the targeted analysis that we were supposed to do, it took more than we expected. So we have done kind of succeeded with the first part of the project very well, but we should have had more time to analyze the data, to get more into the comparative work, the comparison across species. But we see now, and there are probably 23 papers out from the project, but there is probably twice that number in process, and a few of them are focusing the comparative part across salmonids, carp versus zebrafish, and also a comparative study in flatfishes, including turbot. So we have been able to generate and analyze functional annotation maps for both healthy and immune-activated states. We have used the data to predict functionality in genetic variants related to diseases. And we have developed unique tools for comparative work. I didn't touch much on that, but we also have put quite a lot of focus on engaging industries, parties, but also politicians and the general public to get more acceptance for these new tools and get a better understanding of what it can be used for. Okay, so the last slide is just to give a great thank to the consortium. We had 14 academic partners and 10 industry partners in this consortium, and it was really a very good consortium, very good atmosphere, and really good teamwork in this project. So thanks to all of them. Thank you.
Thank you, Sig. Are there questions to our speaker?
Crystal clear.
Michelle?
I have just one provocative question. You said we have progressed in annotation so that we will be able to improve genomic selection. And just this morning, we could see that improvement in prediction accuracy or this or that was not so striking once you were using annotation. And then people were telling maybe this is because of this or that possible explanation. So do you think that your results will still, you can still tell to the breeders it will improve your breeding program?
Yes, so what maybe is different with the focus we have in AQUA-FAANG is that we want to develop tools to avoid challenge testing for diseases, you see? To avoid that, to reduce it, then you need some tools that work across populations and generations. And that's different from doing it with close relatives. And using this deep learning technology, we have shown that in data in AQUA again, that it really works better than just taking a random set of SNPs. So doing the functional predicted SNPs will work better across year classes, and that is in aquaculture kind of slightly different populations. It's not totally different, but they are not directly related.
There's one more question from...
Yes, so thank you. I would like to know how the breeding company are dealing with the negative correlation between production and disease-related threats, for example.
Yeah, so that's of course also in fish species a potential problem. So that we haven't really looked into in this project. So I would say AQUA-FAANG was a project focusing on diseases.
But do you have evidence, for example, for functional variants that are increasing some really nice production trait but have negative effect on other health?
Yes. So that could very well be, but I think also the species we are talking about here is not species that we have been working on that level for a very long time. But you know, they have increased the growth quite a lot in salmon that I know quite a lot of, and that was the selection goal for quite a long time. And it's just until recently they have really started to put a lot of weight into diseases. Yeah. But that's now is the problem in aquaculture is to have a license to produce. So if you don't have a concern about welfare and health, you are not allowed to produce salmon in Norway. And these are infectious diseases, but also parasites like sea lice. So it's becoming more and more important.
Thank you, Sig. I think for the